Most people hear the word CRISPR and immediately think of molecular scissors. That description is not wrong, but it is far too limited. In reality, CRISPR has evolved into a versatile biotechnology platform with many different phases. Part of bacterial immune defense system where it helps bacteria recognize and defend themselves against invading viruses. Scientists later adapted this natural system into a programmable tool for biology and medicine. Its fame came from its ability to cut DNA at a chosen location, which is why it was first introduced to many people as a gene editing technology. That classical role remains incredibly important, but it is only the beginning of the story. Genome editing approach, a guide DNA directs a Cas protein to a specific DNA sequence. The Cas protein creates a break in the DNA, and the cell's own repair machinery repairs that break. Depending on how the repair occurs, the result may be a mutation, a corrected sequence, or a small insertion or deletion. This simple but powerful concept transformed genome editing and established CRISPR as one of the most influential technologies in modern biology. Technology is focused on increasing precision. Base editing allows one DNA base to be converted into another without creating a full double-strand break. Prime editing goes even further by enabling small insertions, deletions, and substitutions with remarkable accuracy. Together, these technologies make CRISPR far more flexible than classical DNA cutting alone. But CRISPR is not only an editor. It can also regulate gene expression without changing the DNA sequence itself. By using modified CRISPR systems, researchers can switch genes on or off in a programmable manner. In other words, CRISPR can function as a gene control system, not just a gene editing system. Another important development is epigenomic editing. Here, the DNA sequence remains unchanged, while the chemical marks that regulate the gene activity are modified. These changes influence whether genes become active or remain silent. Instead of rewriting the genetic code, CRISPR here changes how the cell reads that code. Some CRISPR systems work on RNA rather than DNA. This shifts CRISPR from genome editing to transcriptome engineering. And since RNA molecules are temporary, RNA targeting often produces reversible effects. That makes it an attractive option when permanent genome editing is unnecessary or undesirable. The newest CRISPR technologies continue to expand the boundaries of genome engineering. Researchers are developing systems for mitochondrial editing, CRISPR-associated transposases, and other technologies that are capable of inserting much larger DNA sequences. CRISPR is no longer a single editing method. It's becoming a modular platform for biological engineering. Why is this evolution so important? Because different biological questions require different molecular tools. Sometimes the goal is to permanently modify DNA. Sometimes the objective is simply to regulate the gene activity. Other situations require editing RNA or modifying the epigenetic landscape. Modern CRISPR provides a growing toolkit capable of addressing all of these challenges. So, CRISPR is no longer just a molecular scissors. It's a programmable biotechnology platform capable of editing DNA, regulating genes, modifying epigenetic states, targeting RNA, and much more. The original CRISPR story was about cutting. The modern CRISPR story is about precision programming. Genome editing was only the beginning. The many phases of CRISPR are still expanding.